Today in this video, I'm going to be introducing you to two new concepts, incomplete dominance and codominance. So far in our genetics unit, we've been focused on understanding how organisms have chromosomes made up of genes that code for specific traits. On the two chromosomes that an organism has, each chromosome has an allele that represents for the specific trait. We've mostly focused on the idea of one allele controlling for one trait, although in more complicated genetic theory, it's actually a combination of alleles that controls for complicated traits, such as a person's height or skin color, hair color, etc. So an actual genotype for complicated traits could look something like this, with lots of different alleles represented to show the whole trait. But for 8th grade, we don't have to get into anything quite that messy. We've also focused on the idea of dominant and recessive alleles. For example, here are alleles for a widow's peak in this pair of chromosomes. One chromosome has the dominant allele and the other has the recessive. For normal dominance, as we've been studying it, if a person has the dominant allele represented here by capital W, they will have that trait, even if their other allele is recessive, just like is shown in this chromosome. The dominant allele wins out, and this recessive allele just doesn't get to be expressed unless you had two recessive alleles. Then you would have no widow's peak because that allele, that trait, would get to be expressed. But anytime that you have the dominant trait, it wins out and it's expressed over the recessive trait. At least that's what we've been studying so far. Just when you thought you had the whole thing down, there's always things, of course, that don't follow the rules. The two new concepts are incomplete dominance and codominance. These are ways that alleles express themselves when one allele basically refuses to be recessive. It refuses to not be expressed. So instead of having normal dominant and recessive, you have a different relationship. We'll start with incomplete dominance. In this case, when an organism is heterozygous, instead of the recessive trait not being expressed, the two traits blend together and you get a new third option. The easiest example of this is the flower of the snapdragon plant. Here you have option red, white, or pink. Here's a Punnett square for the snapdragon. In this plant, the dominant flower is red. I'm going to express it with a capital F for flower, and the recessive is white. So if I cross it, here's red crossed with white. All of my genotypes that result are going to be heterozygous. The trick is, though, with incomplete dominance, the heterozygous genotype does not end up being red like with normal dominance. Instead, it blends and you get pink. Here's the same example crossed again, but this time with different letters representing the allele. And you'll notice that these letters are all capital. There is no lowercase. Often when scientists are showing incomplete dominance or codominance, they use all capital letters because nothing really is recessive, so they don't have anything be lowercased. So in this example, they've used capital R to represent red and capital W to represent white. Every cross gives a heterozygous genotype, but with incomplete dominance, that heterozygous genotype leads to a blended phenotype. So you get pink, not just red, pink. The next is an example of codominance. Whereas incomplete dominance is blended, codominance is mixed. Our example for codominance is a roan cow. Here in this example, we have a cow with a reddish coat. It'll be big R. and a cow with a white coat, big W. When you cross the two parents, which the alleles happen to look just like the flowers, but R and W is for a cow now and not flowers. But you get all heterozygous results when you cross the red cow and the white cow. If this were incomplete dominance, you'd get a pinkish cow. But this is complete dominance, so it doesn't blend, it mixes. And you end up getting a spotted cow, which is what a roan cow is. Here you can see the roan cow represented in the Punnett square, but I also want to show you something else you'll often see with codominance. If I use capital C to represent the cow, sometimes you will see the alleles represented like an exponent. So it'll be capital C with this capital W and capital R's up almost like exponents. And then when you cross, this is what it looks like. So sometimes scientists will use this exponent type look for alleles when you're doing codominance. Don't get confused though. It's not like math exponents in the sense that you're not squaring anything. It's just kind of that it's written up high and smaller. So it looks like an exponent. But it's just another way that you'll see codominance represented in Punnett squares and genotypes. So to review, incomplete dominance and codominance only affect an organism's heterozygous genotypes, when the two letters are different. If something is homozygous dominant, so capital R, then it's just dominant like always, or homozygous recessive, capital W, it's still recessive, it doesn't change. But if it is heterozygous, then when it's incomplete dominance, the phenotype is blended, like a red flower crossed with a white flower makes pink. And for codominance, the phenotype is mixed, like a red cow and a white cow making a spotted cow. So that's how incomplete and codominance works in genetics.